what you see here is a measurement of the color intensity using of the OIV system. We have component A and B, and A is the red component of the color, while B is the yellow part. The red cross that represents hybrid A had the higher color intensity, more red than yellow in that hybrid. So you still have the formation of purplish pigments, and this is a marker of the creation of stabilized pigments due to an ethylic bridge in between anthocyanins and tannins. What we obtain is a difference in the stabilization of the color that you can see with these purplish nuances of the color. And then B and C apparently are less stabilized when compared to A, which has the more purplish color. Then things changed after bottling, and this brings us back to the previous graph. Specifically, hybrid A and the control, or hybrid 1 rather, do not change, while the parent and hybrid C shift towards more yellow, more red, which means that there is a sort of decay of the color and less color stability. And here we have analytical indexes that show the reactivity of tannins. These are two analyses whereby we use the reaction of the tannins with a protein in the case of the gel index of serum. And the higher this parameter, the higher the reactivity vis-a-vis or towards proteins. Here we see differences again. The two parents have opposite reactions, which means that when tasting the wines or the tannins, you can perceive fairly different things, and this also increases in the gel index. This also entails a reaction, the tannification of the properties in relation to oxygen and to fining and aging. So we have more reactive tannins when compared to parent 1. In terms of what happens during the tasting, we used here our blind tasting traditional approach with a different order and sequence for each taster using the Latin square design, which means that it's a sort of double-blind tasting. And you also have a Tastel software data processing and a complete statistic treatment, which is what we normally use at the agricultural chamber. The first analysis that I would like to offer to you is the analysis of the principal components. On the first axis, the first graphic on the left, we have more aromatic wines with a tendency to have a higher score, and on the right-hand side, a harder finish, more astringent, bitter wines, and less aromatic in the mouth.
Now, when you look at the bottom quadrants, you have ripe fruit. On the bottom, fresh fruit. On the top, you have ripe fruit. And here's the result in terms of the tastings. You start to see a separation on the first axis between some of the hybrids, among the hybrids more generally, because hybrid B seems to have a more astringent and bitterish finale and less aromatic astringency, while C seems to have a more enjoyable finale with a higher aromatic intensity. Hybrid A is intermediate, and it's similar to the control and the two parents. This is particularly important in understanding the evolution from ripe to fresh fruit. Here are specific criteria that are evaluated when tasting. Color intensity, number A, hybrid A is the most intense, and hybrid B is less intense in terms of color. When it comes to aromatic intensity, it is on the palate that you tell the differences with parent 1 and hybrid C that are in the top quadrants. In terms of the perception on the mouth, you see enjoyability and tannin compared. Hybrid C seems to stand out very clearly with a much more marked minerality compared to the parents on the left-hand side. Now, when it comes to the other sample, you have parent 1 and C, which are less rich in tannin, and they're rounder in terms of tannins compared to the parent and compared to the control. To represent all these nuances and differences, we have developed at the Agricultural Chamber a representation, a chart that we call the profil sensible, or sensitive profile, that represents each of the samples compared to the control. This is a visual way to summarize all the results of our experiment into one simple chart. Now, in this chart, we kept into consideration the objective, the goal, and that is represented with these color codes. The green says that the result has been attained, while red colors indicate unattained goals. And everything is represented around the central line, which is the control sample, the LC22. When you go to the right, you increase the character. If you go to the left, the character is less evident for some undesirable characteristics that might be the intended goal. Then you have the 4 levels. If the 4 boxes have been filled, the difference is highly significant, 1% error. 3 is significant, 5% error. 2 full boxes, they designate a strong trend, 10% error in statistical terms. And 1 single box is a weak trend with 30% error. And that is to indicate a slight tendency that might emerge, but that should be interpreted with some caution. So in green you have the goal attained, while in red there's an undesirable deviation based on what we were looking for. Some descriptors are in gray, also to clarify that there's a clear-cut difference. Here there's a difference between the parents and the hybrids. Without going into the details, the 2 parents have interesting elements and positive that are represented in green, some with a very high significance, particularly parent 2 when it comes to length on the palate, particularly riper fruit, which in the case of this product wasn't necessarily what we were looking for, but that was nevertheless present. While for parent 1 we have some qualities and some defects, such as an intensity and an aroma on the palate, which are very clear cut, with a slight positive tendency to have a rounder and better structured balance and less astringency compared to the control. When it comes to the hybrids, hybrid A displays interesting characteristics in terms of color, the characters of fresh fruit and complexity, and it's particularly interesting on the palate with a well-structured palate and a good quality of tannins, superior tannins versus the control. Hybrid B has a profile almost entirely in red, so it does not correspond to the goals that we were pursuing. And at the tasting, it becomes evident that it will be discarded. Hybrid C only has green characterizations, green boxes. There are no meaningful, no significant differences, but all positive characteristics, and it seems to point to better quality, better roundness, intense aroma, overall balance, and a very interesting finish with less bitterness and less astringency. So hybrids A and C are selected. These are the Safino HTS 62 and HDS 135 tests. And I'm now going to leave the floor to Etienne, who will continue with the presentation. Thank you very much, Jean-Christophe. I am taking the floor for the third part of the presentation on easy-to-use production, because it's interesting to look at the various technological characteristics, but then you need to be able to produce that too, and it's a different story, and you need to produce the best possible quality. To conclude on what Jean-Christophe just said, we completed a test, and the test results integrating that with over 30 tests for each stock, each strain out in the vineyard. And we evaluated the capacity of HTS 62 to increase the color and improve the quality of tannins, and also to enhance the aromatic potential with fresh fruit notes. And these wines are easier also to sell because of its good fruit. Now, this is the production of yeast. It is just summarily represented with all the various steps in industrial production, beginning with collection, which might be in glycerol at -80 for strains that are not frequently produced, or they can be produced at 4 degrees for the strains that can be produced industrially and therefore also more frequently. The process in itself is a multi-step process. The first steps take place in an anaerobic and aerobic environment, almost in terms of alcoholic production, with the minor alcohol production to be able to produce the necessary biomass. Now, all of that is done in the labs once the yeast have been grown by multiplication, by reproduction to the necessary extent. The yeast will then be moved into big tanks where the biomass will be continuously produced under controlled conditions while checking oxygen, sugar, vitamins, and all the relevant parameters. And all of that takes place under anaerobic conditions to increase the biomass and reduce ethanol production that would hinder yields and production, which is exactly what we need to obtain through our experiments. Once this biomass has been obtained in the form of a cream yeast, it is possible to preserve it at 4 degrees, and so the yeasts will be preserved in their fresh form, which also has an impact on their life. 2-3 weeks, relatively short period of time for fresh yeast. To improve storage and preservation of cream yeast, a number of further steps, including drying, need to take place. For instance, you can put the yeast through a drying machine. I don't know if you've ever had a chance to see compressed cream yeast bars, and once you obtain that 35% drying, it is possible to extrude it, to draw it in the form of spaghetti, to dry it even better and obtain better final results and obtain the dry yeast. Now, in this yeast production process, there are a number of essential parameters. These are pure cultures. You can't mix strains, at least not initially, during the production cycle. And during production, you have to look for the maximum growth rate, controlling alcohol production, which is connected with the consumption of carbon and sugar, air, and temperature in the substrate. The pH also needs to be controlled, because a very low pH will control contamination, but it also might lead to coloring of the material. Temperature will also to be monitored to guarantee proper yeast activity, and most importantly, nitrogen. In industrial terms, there's just one recipe to produce each strain. Each strain has different characteristics and has its own specificities. However, the multi-stage process is the same for all. During this industrial scale production, genetic stability tests will be run to understand exactly what strain we're talking about, and production suitability tests and industrial dehydration suitability tests as well. These are essential in order to produce biomass. So industrial production can rely on a relatively high growth rate to have a reasonable yield. And then the second important parameter has to do with this. With this yeast, with this procedure, we can grow all strains, but they have to be preserved and stored, because otherwise there will be problems in the production. So this is a very specific industrial know-how based on reproduction, as well as on the physiological stage at which the yeast will be stopped during growth. This is a schematic representation of the 4 stages in yeast growth, with a G1 gap, which has to do with the growth of the yeasts until they reach the critical mass before going to stage S, where the genetic material will be replicated. Then you go to latency in preparation for mitosis, which will take place during the stage called M here, in order to obtain a true duplicate of the yeast and have yeasts that have exactly the same genetic materials and contents, just replicated. The role of the expert is to stop production at the best physiological stage, so that the yeasts can be dried under the best humidity conditions. Let's see what happens at a replication level. There's a germination. If you stop growth at that point, you know for sure that the life of the yeast will not be something that will last long term, because the membrane will be fragile. So this is indeed a very interesting stage of the growth, and not just for physiological reasons connected with the membrane, but that we should stop at a stage when the yeast has all the doors wide open to absorb everything from the substrate, the sugar, the carbon in the substrate. And it is immediate, instantaneous absorption, because whenever there's some amount of sugar available at that stage, it will start to absorb even without going through latency. So it's very important to understand at what point we needed to stop yeast reproduction to dehydrate. Now, dehydration needs to factor in 2 important factors. The preservation for dehydration and rehydration, and also the fats. It's also important to use a certain recipe for production. Lipids or fats are very important, as well as trehalose. This is very interesting because it has a role in terms of the way the operation of the membrane. As you can see, there are phospholipids constituting the membrane. When the yeast is rehydrated in the fluid form, and there's a crystallization during dehydration that can cause crystals and involve damage. Of course, when you dehydrate, the yeast will not be able to be rehydrated appropriately. While with the addition of trehalose, the structure will be preserved. It's a relatively fluid structure that will be maintained nevertheless despite dehydration. Of course, as you know, we generally add an emulsifier, a plant oil, in order to improve the yeast resistance when dehydrating. During dehydration, we use a dehydration system which is as soft as possible, a fluid bed generally. We will try to make the filaments of the yeast more fluid, and that using a very soft approach in order to avoid these risks. Dehydrating the yeast, the membrane will fold onto itself, and we need to avoid the creation of crystalline structures that might create vesicles that, when rehydrating, might break the membrane with a huge risk. So this is what you need to know in industrial terms when it comes to the reproduction and the effects of dehydration. The easy-to-use process factors in all these parameters, and we have in the course of time validated a certain number of strains. The easy-to-use process is specific for yeast production and makes it possible to produce dehydrated yeasts that can stand different handling conditions and different conditions of use. The validated strains are the following: the SC22, which was the previous control, the NDA21, and the others listed here. So in order to connect to the development of the hybrids that we have developed, we selected the hybrids based on their fermentation and sensory characteristics, not the analytical characteristics. And it is interesting to have these strains, which are also easy to use, and they can be produced in an easy-to-use form according to three parameters: the viability of these yeasts after rehydration in the different conditions of use, their analytical results, and sensory results. Now, if you put the yeast in water, just tap water plus sugar at different temperatures between 10 and 43 degrees C, in either water with 25% sugar, just to simulate the natural conditions, and then a number of variations. And if you look at the viability after rehydration, it's very interesting. This parameter gives us, has given us many results on all the validated strains that point to the fact that when you rehydrate all these strains, the viability is still very high, and it is already very good. Now, viability is good, but if you have a broken leg or a broken arm, it's harder to work well. So at that point, we had to look at a different parameter, which is the fermentation performance, to validate that viability. Among the various conditions, in this case, we had three that we needed to meet, which might be relevant for work at the winery. They're listed here. Usual conditions of use: initially rehydration with progressive acclimatization to the must with additions of must before inoculation, and then that is moved to the tank, and then rehydration in cold conditions. You take cold tap water at 15-17 degrees, just fresh cool water, and then you transfer it before inoculation. Or you can prepare it with the must, with direct inoculation in the must that needs to be fermented, and then that is moved directly into the tank, the fermentation tank. All these parameters were applied on this Malbec sample with an Argentinian research and development partner. And here's what we obtained from a kinetic point of view on these two strains, S135 and S62. The curve at the top is the temperature curve, so you start with... Now, if you look at the different positions, there are no differences in kinetic terms. Then we carried out statistical analysis on the analytical parameters, and there are again no meaningful differences between direct inoculation and inoculation through acclimatization. Fermentation performance. It was associated with the sensory profiles using triangular tests, and these are basically the results when compared to the customary method versus acclimatization. There are no meaningful differences between the two strains if you use the usual conditions. So we also evaluated that from an analytical point of view, and this procedure, particularly the direct inoculation in must, is relatively cost effective and makes water unnecessary. So that saves time as well, which makes it pretty interesting. So it's interesting to inoculate it directly before rehydration, and that is very relevant for the people working in wine production. So in conclusion, hybridization is very powerful, but it's nothing if you don't screen specifically for the intended objective, particularly in terms of polyphenol extraction and fermentation characteristics. And then also the industrial competencies are essential in order to produce the best quality yeast. And the processes developed by the group, the EASY-2-USE process, tell us that it is possible to preserve the fermentation characteristics from an analytical and sensory point of view, which are specific by strain, while simplifying also its use, which might also involve major savings in the years ahead. Thank you very much for listening so attentively.